Today let's discuss about dicot stem and monocot stem, that is anatomy of dicot stem and monocot stem. So the outermost layer is the epidermis. It is made up of closely arranged parenchymatous cells. So epidermis is protected by an extra layer called cuticle. Cuticle is mostly made up of wax. Since it is waxy in nature, it will prevent the excess loss of water from the stem, as well as it will protect the stem from getting soaked during excess rain. And from the epidermal cells, some multicellular extensions called trichome will arise. And the main purpose of this trichome already we have seen. It acts as a defense agent as it secretes some chemicals, or due to its stiff nature, it is considered as a defense agent. So here the layer between epidermis and pericycle constitute cortex. So all the layers below epidermis and pericycle forms the cortex, and cortex here is divided into 3 sub-layers. So what are they? Immediately below epidermis, few layers of coloncymatous cells will be present. So coloncymatous cells have thick wall, and it actually give strength to the stem. So immediately below epidermis, few layers of coloncyma will be present. Coloncyma are thin, thick-walled cells, and it gives mechanical strength to the stem. And this layer which has this coloncyma cells are known as hypodermis. So next to hypodermis is loosely arranged. I mean the cells will be arranged here and there with intracellular spaces, and is made up of thin-walled parenchymatous cells, and this layer constitutes the cortical layer. So next to the cortical layer is closely arranged barrel-shaped endodermis. So cells will be barrel-shaped and they are closely arranged, but here these cells have starch in it, and this layer is hence known as starch sheath. Each cells have starch. Each cell of endodermis have starch in it, and hence the endodermis is together known as starch sheath. So the first layer is epidermis. Below the epidermis, the cortex is divided into 3 layers. The first layer being the coloncyma, being the hypodermis, which is made up of thick-walled coloncyma cells. The coloncyma cells basically give the mechanical strength. So next to hypodermis is the loosely arranged parenchymatous cells with intracellular spaces among them, and this layer which has the parenchyma cells form the cortical layer. So next to the cortical layer is the tightly arranged barrel-shaped cells which contain starch. This layer constitutes the endodermis, which is also known as starch sheath. So when you come next to endodermis, you have pericycle. So the cells of pericycle are also closely arranged, but here the cells are made up of sclerenchyma. So sclerenchymatous cells are very thick. The walls will be very thick, and it mainly acts as it mainly gives strength to the part in which it is present. So next to pericycle lies the vascular bundle. So in the center, when you take the stem, in the center you can see parenchyma cells arranged in the form of rays, and these rays constitute the medullary rays. In the center, again loosely arranged thin-walled parenchyma cells with intracellular spaces will be present, and this layer is known as pith layer. So this is the vascular bundle of a dicot stem. You can see they are arranged in the form of a ring. So this is a identical feature of this dicot stem. The vascular bundles will be arranged in the form of a ring. So already you have seen this kind of vascular bundle is known as conjoint vascular bundle. Here both xylem and phloem occur together. In the center, cambium is present. So when cambium is present, secondary growth is possible, and hence it is a open vascular bundle. And you can see the smaller xylem, protoxylem, the first formed protoxylem will be facing the center, and the metaxylem will be facing outward. When the protoxylem is facing the center, it is known as end arc protoxylem. So here the vascular bundle is arranged in the form of a ring, and each bundle is a conjoint vascular bundle as xylem and phloem occur together. Since they have cambium in the center, it is capable of doing secondary growth, and hence the vascular bundle here is open. The last thing is the first formed protoxylem will be facing the center, whereas the later formed metaxylem will be facing outwards, and this kind of arrangement is known as end arc. So what about monocot stem? Epidermis is similar. So when you take hypodermis, instead of collenchyma, here sclerenchyma cells will be present. Since sclerenchyma has very thick wall, it acts as a strength giving structure. And here the vascular bundles are scattered. It is not arranged in any manner. It is scattered here and there. And in this, if you see, it is a conjoined. Again, xylem and phloem are occurring together, so it is conjoined. Since it is a monocot, no secondary growth will be there, and hence it is cambium will be absent, and hence it is a closed vascular bundle. Here also the vascular bundle is conjoined, but cambium is absent, and hence it is a closed vascular bundle. And this outer peripheral vascular bundles, if you see, they are smaller when you compare to the center vascular bundle. This is also an identical feature. And in the phloem, actually some cells will be present. In the phloem, parenchyma cells are absent. And if you take this, so this is the monocot vascular bundle. You can see white cavities here and there. Cavities are empty spaces, but here it is filled with water. So water containing cavities are present in the vascular bundles, and this is also an identical feature of monocot stem.